Welcome to this discussion on the ICH Q1A R2 guideline, stability testing of new drug substances and products. This guideline, updated in February 2003, provides a harmonized approach to stability testing for pharmaceutical registration across the European Union, Japan, and the United States. What are the core objectives of this guideline?
The primary objective is to define the minimum stability data package needed for registering a new drug substance or product in the three ICH regions. It aims to provide a consistent framework while allowing flexibility for specific scientific considerations. The goal is to ensure the quality, safety, and efficacy of pharmaceuticals throughout their shelf life.
The guideline mentions climatic zones. How do these zones influence the testing procedures?
The guideline acknowledges that climatic conditions vary globally, impacting drug stability. It focuses on climatic zones I and II, representing the conditions in the three ICH regions. The principle is that stability data generated in one region should be acceptable to the others, provided the testing adheres to the guideline.
The guideline details different storage conditions: long-term, intermediate, and accelerated. Can you explain the rationale behind these different conditions?
Long-term testing simulates real-world storage conditions to determine the actual shelf life. Accelerated testing uses exaggerated conditions, higher temperature and humidity, to speed up degradation, allowing for quicker shelf life estimations. Intermediate testing bridges the gap, providing data between long-term and accelerated conditions.
What are the key parameters considered during stability testing, and how are they evaluated?
Key parameters include assay, drug content, degradation products, physical attributes, appearance, pH, and microbiological properties where applicable. Evaluation involves analyzing changes over time, often using statistical methods to determine the time when the product no longer meets its specifications.
The guideline mentions significant change. How is this defined, and what actions are taken if a significant change is observed?
Significant change is defined differently for drug substances and products. For drug substances, it's simply failing to meet the specification. For drug products, it's more nuanced, involving changes in assay, degradation products, physical attributes, and other relevant parameters. If significant change occurs during accelerated testing, further testing under intermediate conditions is usually required.
The guideline discusses bracketing and matrixing as ways to reduce the number of tests. What are these approaches?
Bracketing involves testing only the extremes of a range, VG, highest and lowest drug concentration, assuming intermediate levels behave similarly. Matrixing tests a subset of samples at each time point, extrapolating to the rest. Both approaches aim to reduce testing while maintaining sufficient data for reliable conclusions.
How does the guideline address the issue of establishing a retest period for drug substances and a shelf life for drug products?
The guideline outlines procedures for determining both. It emphasizes the importance of testing at least 3 batches and using statistical analysis to project the retest period or shelf life. Post-approval commitments to continue stability studies may be necessary if initial data don't cover the entire proposed period.
The guideline mentions supporting data. What kind of information falls under this category?
Supporting data includes information beyond the formal stability studies, such as data from early batches, different packaging, or other relevant scientific rationale. This helps to build a comprehensive picture of the drug's stability.
What is the significance of the glossary provided in the guideline?
The glossary is crucial for ensuring consistent understanding of terminology used throughout the guideline. It defines key terms like accelerated testing, bracketing, climatic zones, and many others, preventing ambiguity and ensuring everyone is on the same page.
The guideline references other guidelines: Q1B, Q1C, Q3A, Q3B, Q5C, Q6A, Q6B. Why are these cross-references important?
These cross-references highlight the interconnectedness of different aspects of pharmaceutical development and regulation. They direct readers to related guidelines for more detailed information on specific topics, such as photostability testing, Q1B, or impurities, Q3A and Q3B.
How does the guideline address the specific challenges of drug products packaged in semi-permeable containers?
For products in semi-permeable containers, the guideline emphasizes the importance of evaluating potential water loss, especially under low humidity conditions. It provides specific storage conditions and methods for assessing water loss, including calculations based on water loss ratios at different humidity levels.
What are the implications of the guideline for drug products intended for storage in refrigerators or freezers?
For refrigerated products, the guideline recommends long-term testing at 5 degrees C, but only 3 degrees C, and accelerated testing at 25 degrees C, NAMWALA 2 degrees, 60%, RH Neller and 5%. For frozen products, shelf life is based on real-time data at negative 20 degrees C, negative 5 degrees C, with additional testing at elevated temperatures to assess the impact of short-term excursions.
In summary, what is the overall impact of the ICH Q1A R2 guideline on the pharmaceutical industry?
The guideline provides a standardized, internationally harmonized approach to stability testing, streamlining the drug registration process and ensuring consistent quality standards across different regulatory agencies. This reduces redundancy and facilitates global trade of pharmaceuticals. This discussion has only touched upon the key aspects. A thorough review of the guideline itself is recommended for a complete understanding.
Thank you for this informative discussion.